Hello. Oh, wait. Good morning and welcome to the lecture series on clinical trial readiness for syndromic neurodevelopmental disorders. We are delighted that Dr. Shefali Jeste accepted the invitation to present today. Before I officially introduce our presenter, please allow me to address a few logistical items. The Tarjan Center at UCLA is a federally designated center for excellence in developmental disabilities and your host for today's lecture series. We'd like to share some information with you regarding today's session so that you can participate as fully as possible. For today's lecture series, we will be recording the lecture, but at the same time, there will not be an opportunity for you to necessarily speak during the lecture. We've muted all calls. As we proceed through the presentation, our presenter would love for you to provide as many questions as you have. In order to do that, I'm asking you at this time to please look at the bottom of your Zoom program display where you will find a chat box or a text box. We're asking that you insert your questions into the chat box throughout the duration of the lecture series. We will address your questions at the end of the presentation, but we're asking you to insert your questions as they naturally arise as you're listening to the presentation. The other thing that we'd like to share with you is that there's a closed captioning feature to this webinar, and you may use that closed captioning feature by looking at the bottom of your Zoom display towards the center of the screen and by clicking on closed captioning and enable that function for you. In addition to that, there's a service that is being provided through which you can increase the font size of the closed captioning, and you may do that by using the website streamtext.net, streamtext.net. In addition to that, towards the end of the lecture, we will be inserting an evaluation link into the chat box. We're asking that you click on that link and complete the evaluation. The evaluation is important because it will help us as we develop additional content for training such as today's lecture. Thank you, and without further ado, here's Dr. Elizabeth Lawgason that will introduce today's speaker.
Well, good morning, everyone. On behalf of the Tarjan Center and our co-sponsors, the Center for Autism Research and Treatment at UCLA and the UCLA Intellectual and Developmental Disabilities Research Center, the IDDRC, I'd like to welcome you all to our monthly lecture series and our second lecture series over Zoom. We appreciate your attendance at this lecture, particularly given these really uncertain times, and we hope that you and your family and friends are staying safe. And we hope it won't be long before we're all able to meet in person again. With that said, it's my pleasure to welcome my friend and my colleague and our distinguished speaker, Dr. Shefali Jeste. By way of background, Dr. Jeste is a behavioral child neurologist specializing in autism and related neurodevelopmental disorders. She's an associate professor in psychiatry, neurology, and pediatrics in the UCLA David Geffen School of Medicine. She's the director of the UCLA Caring Clinic and the director of the Biomarkers Core of the UCLA Center for Autism Research and Treatment. After earning her BA in philosophy from Yale University in 1997 and her MD from Harvard Medical School in 2002, Dr. Jeste completed a residency in child neurology and a fellowship in Behavioral Children's Hospital, Boston. She then pursued postdoctoral training in developmental cognitive neuroscience with Dr. Chuck Nelson at Harvard Medical School, where she gained expertise in the use of high-density EEG to characterize functional brain development in infancy and early childhood, particularly as it informs atypical development. She was then recruited to UCLA in 2010 as the director of the UCLA CART Biomarkers Core. Dr. Jeste's research is focused on developing methods to improve our precision in the diagnosis and treatment of neurodevelopmental disorders through the application of brain-based biomarkers and more targeted developmental phenotyping. Her lab studies neurodevelopment from early infancy through late childhood, and she is the principal investigator on numerous studies. Dr. Jeste holds many national and international leadership positions, including the board of directors of the American Brain Foundation and the board of directors of the International Society for Autism Research. She also serves as the chair of the International Baby Research Consortium. And last year, she's won many awards, but last year she was awarded the 2019 Presidential Early Career Award, which is the federal government's highest honor recognizing contributions to science. So we are very proud of her for that distinction. Today, Dr. Jeste will be presenting what I'm sure will be a very interesting lecture on clinical trial readiness for syndromic neurodevelopmental disorders. So please join me in welcoming Dr. Polly Jeste.
Thank you, Liz, and all of you who are attending today and listening. I really appreciate the invitation. And it's always harder to give a talk when I can't see my audience, but I did take a quick look at the participants, and I see it's a really nice range of students, research coordinators in labs, as well as faculty members. And so because of the range of backgrounds that are in this audience, I am going to try to keep the discussion at a fairly basic level with maybe some higher level data slides and concepts thrown in. But if there are times where you really need clarification, I will try to see if I can see a clarification question in chat. Otherwise, I'll definitely at the end. I'll try to, brevity is never my strong suit, but I'm going to try to finish with at least a few minutes so I can answer questions, and you all can also email me afterwards. So today I'm going to actually talk about what I like to think of as clinical trial readiness for syndromic neurodevelopmental disorders. This is a really exciting time in our field where our genetic testing, as you'll see is my theme from this talk, our genetic testing is not just academic anymore. We actually have actionable items and goals that we can really pursue through genetic testing. And that's just been a really big breakthrough in our field over the last few years. And so I'm going to talk to you a little bit about that today. And so we'll kind of get started. And again, as I said, I'm sorry I can't see all of you in person, but I really appreciate your taking the time to listen and learn. So just disclosures, I do some consulting for Roche, but actually nothing I'm going to talk about today is relevant to the consulting work that I've done for industry. So what I'm going to do is I'm going to start by talking for about 10 minutes, just give you an overview of how we think about syndromic neurodevelopmental disorders, and really what genetic testing what kind of doors genetic testing has opened for us in neurodevelopmental disabilities. I'm then going to spend the bulk of my time talking about this concept of clinical trial readiness, meaning, what does it mean to be ready for these trials as they're coming up? And what can we do? And I'm going to show you specific data that we've acquired from my lab and from collaborations with others in the space of clinical trial readiness, really just to show you examples to get you thinking about, again, how the field is moving towards being ready for trials. And then we'll end with some next steps. I'll show you some exciting studies that are ongoing right now at UCLA in case you're interested in learning more, being involved. I did want to say that I am leaving my video on for now, but my internet has been a little bit unstable today. I've already been booted off of Zoom 4 or 5 times in my own lab meeting this morning. So if I get any alerts on my computer that my internet is slowing down, I'm just going to shut my video off. so that you can just see the slides and don't have to see me, because I think it'll speed things up a bit. Okay, so let's start. So as I'm sure you know, there have been a lot of advances over the last decade to 2 decades in how we examine genetic makeup of any patient. And here I say autism genetics, but really in any patient. We used to really just have the technology to look at the karyotype and to identify whether there were extra numbers of chromosomes, but that's transitioned very quickly into chromosomal microarray technology, which allows us to look at and identify areas of the chromosome that are either duplicated or deleted, and those copy number variants that we detect contain many, many genes. And then at an even higher, more sophisticated level, we can now use whole exome and whole genome sequencing to identify mutations in single genes and single base pairs that lead to mutations, again, in individual genes. And with that technology, what's happened that over again the last, and this is just a slide showing specifically autism genetics, but again, I would really think more broadly about this as neurodevelopmental genetics. What we've started is a lot of different syndromes where we're finding patients that have individual, again, gene mutations or copy number variant syndromes, as well as more traditional syndromes like tuberous sclerosis that have been around for a long time. We're identifying these specific genetic etiologies that explain the neurodevelopmental disorders or disabilities that we're diagnosing in our clinics. And based on just the availability of that technology, what's actually happened is that now genetic testing is really routinely recommended as the medical workup for any individual with ASD or autism. And again, I just want to really emphasize that you're going to hear me talk about autism quite a bit here, because many of the guidelines and many of the studies have focused on autism. But really, as you'll see throughout this talk, we're really talking more about the broader neurodevelopmental disability spectrum. So kids with all kinds of different delays or challenges in their developmental abilities. And we'll talk about that more as we go along. But really, again, this is the routinely recommended medical workup. And the recommendation now is that any child, infant or child with an ASD should undergo chromosomal microarray testing. Boys, it's still recommended that they have testing for Fragile X, while girls are recommended to have testing for MECP2 sequencing, which diagnoses Rett syndrome and related conditions. And then in some centers like ours, which we'll talk about in a second, we are moving on if that testing is normal or not suggestive of an etiology, that we move on to the more precise whole exome sequencing. Now, whole exome sequencing is not routinely performed in all centers, partly because of the cost and the availability, but there have been some studies recently that have shown that it is quite high yield. This was a meta-analysis that was published just last year, looking at the yield of whole exome sequencing in patients with neurodevelopmental disorder, like a classic kind of forest plot. And what you see is that especially in conditions, in individuals who have neurodevelopmental disorders plus other comorbidities, the yield of exome sequencing was quite high, up to 40 to 60%, meaning that in patients that had this testing, 40 to 60% were actually found to have a diagnosis when exome sequencing was performed. And in that particular analysis, they actually went on to recommend that any patient who has either intellectual disability or autism, regardless of whether they have a more syndromic presentation, which we'll talk about, that they should undergo both whole exome sequencing and the chromosomal microarray. Now, again, that's a great recommendation. Part of the challenge, again, is that whole exome sequencing is quite a bit more expensive and not as easily accessible. And so we do sometimes face that challenge. And even in our clinic, which I'll mention in a little bit, we are not often able to obtain the whole exome sequencing on every patient. But I think that will be changing over time, especially as insurance companies become more aware of the fact that this testing can be quite high yield. But regardless of this, what we have found overall, based on the clinical availability of all this testing and many studies that have actually just examined genetic etiologies in patients with neurodevelopmental disorders, is that about a quarter of patients with intellectual disability or autism have an identified genetic cause. So that's a pretty substantial number, and a much bigger number than what we were quoting 10 years ago when we first were really using these technologies more proficiently in the clinical setting. So we have that, we have that statistic that I just told you, but when we see patients in clinic, this is the question I'm always asked by patients. And again, if I were looking at all of you in the audience, hopefully I'd get some head nodding right now from those of you who've heard patients ask this exact question. Maybe not so much for the geneticists, but certainly for us in pediatrics or clinical neurology, which is, well, okay, fine, you want to get genetic testing on my child, but why? So isn't it just academic? Are you just going to learn about cause? How is that going to actually help my child or my family? And I will say, honestly, 15 years ago when I was a trainee at Boston Children's, I feel dated now because I used to say 10 years ago, but 10 years ago I was already here, getting old by the minute. But when I was a trainee, we were actually at Boston Children's obtaining microarray analysis on all patients, even back then, just because the hospital had the funds to cover that testing. And most of our results were actually kind of interesting variants, right? Without us really being able to act on or advise families based on that testing. So kind of academic at that time. However, now that is absolutely not the case. And I really emphasize this because those of you who might be in the space of actually assessing children for the neurodevelopmental disability, meaning if you're a clinical psychologist or an interventionist and you're working with these families, you might get asked this question by them as well, right? Because their medical provider might be suggesting genetic testing. So I think having a way to have a dialogue with patients around why is it not just academic can be quite helpful. And so this is what I tell patients, okay? And I'm going to show you examples of the first two, but we're really going to focus on bullet point number 3 in the space of clinical trials. So the first is that genetic testing has really opened the door for community building or linking families to more specific communities, right, that are based on the specific genetic etiology that their child has. And I'll show you an example of that in a minute. The second is that as we find out about the genetic etiology and we gather more data on more kids or adults with the same genetic or the same genetic mutation, we start identifying clinical patterns, right? So we know that certain syndromes might be associated with certain clinical features, and that helps actually us as clinicians guide the way we will monitor that patient and even talk to the family about what might be expected in the future. And then lastly, and this is really new and super exciting, is that as we identify genetic etiologies, we actually are able to develop targeted therapeutics based on understanding the underlying mechanism that is implicated in whatever the genetic, again, mutation or variant is. And with that, we're opening the door for clinical trials. And again, that's what we're going to focus on today. But I do just want to show you a couple of examples of these first two. Okay, so community building. So what's happened over time is as syndromes are identified, and when I say syndromes, in this case, what I mean is specific genetic etiologies are identified, such as 15q duplication syndrome, which you're going to hear more about in a little bit because that's a syndrome we study quite a bit in my group, Tuberous sclerosis, Fragile X, Phelan-McDermott. As these different genetic causes are identified, what happens is these amazing groups of parents band together and they form PAGs, PAGs, which is patient advocacy groups. And what do those PAGs do? Well, they support families who have new diagnoses. So if a patient newly is diagnosed with a genetic variant, they can go to that PAG's website and immediately get connected to a bunch of other parents who've been through the same experience, either recently or long back. They also have developed both clinical and research resources, right? So they find investigators who might be studying mechanisms of disease that are relevant to the specific genetic cause or variant. And they have research conferences. They fund research grants. They develop national clinics. So for instance, we just became one of the national 15q clinics where we see kids with Duke 15q and Angelman syndrome. And those clinics are sponsored by the Angelman Syndrome Foundation and the Duke 15q Alliance. So they're really, really leveraging, again, the power of this community to promote good clinical care. They do advocacy. They'll go to Washington and advocate for research funding for their specific condition. Tuberous sclerosis has done an incredible job of this. And in fact, the Department of Defense funds millions of dollars a year on tuberous sclerosis research because of families advocating. And then specifically from the standpoint of facilitating research, a lot of these PAGs are developing their own patient registries, biorepositories, as well as really forwarding clinical trials. So this is a really, really exciting area. And I will say that what I'm going to describe at the end in terms of trials that are ongoing are largely promoted and motivated by these PAGs. Recently, in the last couple of years, a group developed that I play a very active role in, and just something for you all to know about, called Agenda, which stands for the Alliance for Genetic Etiologies and Neurodevelopmental Disorders and Autism. That's a big mouthful. But basically the idea of Agenda, and it's actually sponsored by the Autism Science Foundation, ASF, it's a gathering of all the heads of these different PAGs so that the PAG leaders can talk together about these initiatives that I'm listing here, right? Because if you think about it, the mechanism behind, for instance, an effective patient registry is not really specific to the genetic condition. The data that go in might be a little bit specific by cause, but the basic mechanism to get a good registry going and get parents and families to enter data and to have the clinicians engaged, like that process is pretty common across these different rare diseases, these different syndromes. And so Agenda was developed to allow for crosstalk amongst these PAGs. And we've been actually doing quite a bit of work as a group, again, to really think about how we use PAGs to facilitate research. So we did in our clinic, and I'm going to talk more about the clinic in the next bullet point, but in our clinic at UCLA called Care and Research in Neurogenetics, where we see kids with all different syndromes, we actually did a survey and we asked them about their entire diagnostic journey. But one of the things we asked about was support. Where do you get your support, especially once your child was diagnosed with a specific genetic condition? And in our cohort, more than 3/4 of our families reported they were involved with a specific patient advocacy group. And what was really interesting is that those families that were in a PAG were more likely to indicate that they had great confidence in knowing how to proceed steps for care for their child, whereas less than half of the families that were not a member of the PAG, of a PAG indicated confidence in next steps. And so we're seeing that these PAGs are actually really giving parents a solid footing in helping them know what to do next. And I think that's really important, and this is really growing quickly. And I think actually the growing quickly piece is shown by this last bullet that I have here, which is that what we found was that all of our parents of kids under 3 were engaged with a PAG. Less parents of adults with genetic syndromes were associated with PAGs. And I think that's partly because, this is sort of a culture shift. Like these PAGs are just now forming, again, especially with the power of social media and everything else. And so a newly diagnosed young patient, that family is much more likely to immediately try to connect with these groups. So again, really important here from the standpoint of promoting both care for families and research, as we'll talk about. But the second thing I tell parents, when they ask me about why genetic testing, is that from a clinical standpoint, it can really help us with clinical monitoring and thinking about, again, which comorbidities or other developmental challenges may be more specific to that genetic etiology. And I don't have time to go into all the details around this at all, but I will just say that what we know as a whole in patients who have genetic syndromes associated with neurodevelopmental disabilities, again, what I'm calling syndromic NDDs, is that it's not simply autism. They may undergo genetic testing because of an autism diagnosis. But what we know is that children or adults who have genetic syndromes, they're more likely to have kind of autism plus, as we might call it. So they're more likely to have global developmental delay, and I'll show you examples of this in some data moving forward, especially in the motor domain. They're more likely to have intellectual disability, often quite severe. They're much more likely to have neurological comorbidities such as epilepsy. For example, and I'm going to show you data on both of these syndromes, Duke-Patine-Q syndrome, tuberous sclerosis. Both of those conditions, more than 50% of patients with those conditions have epilepsy. So you can imagine in a patient with an identified genetic variant that's highly comorbid for epilepsy, that's a patient that I might think about doing a baseline EEG on just to have some baseline information to rule out subclinical seizures, but also just to know what their EEG at baseline is like, so that if there's a change, I have something to compare it to. In a patient like that, I might also take spells much more seriously, right? So a staring spell, I may, think is more behavioral in a patient who's at low risk for epilepsy, but in a patient with a genetic condition who's having new staring spells, I would be much more quick to obtain an EEG to rule out a seizure. So that's just an example. And then other system involvement. So kids with P10 mutations, for instance, are a higher risk for having a variety of different cancers. And so there actually is a clinic at UCLA that follows these patients just to monitor them for different oncological comorbidities. So things like that. So again, knowing about the syndrome does help guide clinical monitoring. And it's actually in that setting that we did develop this clinic we mentioned in other talks called Care and Research in Neurogenetics. The idea of this clinic was really to have a hub for families of kids and adults with neurodevelopmental disabilities who had known genetic trends.
Dr. Jeste, are you still there?
That. Okay, so again, I hope I've sort of in the beginning, first part of this talk, just given you, some sort of language to use with families around this question about why genetic testing. But really, this is where the excitement comes in, and this is what I think has been, again, what really drives so much of the work that we do right now, which is that we can now tell parents that, hey, we have the potential for some precision therapeutics for certain genetic conditions, and I think this is only going to explode over the next decade just because of the really beautiful, exquisite work being done at the basic, in the basic neurosciences to really understand disease mechanisms. So let's take a step back for a second and think about precision health, right? So what is precision health? One way to sort of operationalize it is that the idea, especially in neurodevelopmental disorders, is that in precision health, you have treatments that are specific to subsets of individuals, and that might be more effective for really getting at cause or mechanism, right? So rather than thinking about, if you think of the whole spectrum of NDDs, it's often ineffective to try one drug or one treatment for the whole group and expect all of the individuals to respond similarly, right? Whereas if you can identify meaningful subgroups, and in our case, genetically identified subgroups, you might then be able to think about treatments that are, again, more targeted for those specific genetic etiologies. And what's happened in genetics is just that. So we have the opportunity for precision therapeutics in these specific genetic conditions. And those precision therapeutics come in different flavors. And I don't have time to go through, every single type of treatment. But, we can think about medications, especially, for instance, if we have, we know the genetic etiology of a certain kind of epilepsy. For instance, someone has a mutation in a sodium channel, then there are drugs that target the sodium channel that might be more effective to treat seizures in those patients. But there's also more like really right at the source of the genetic variant or change. There are treatments now. So there's amazing technology in gene editing using CRISPR-Cas9. And then now there's also, again, the excitement around ASOs or antisense oligonucleotides. And I'll quickly, again, in a very sort of basic way, for those of you who are not biologists in the room, remind you about what ASOs are, because you're going to be hearing a lot about them. And the reason I bring it up is that, again, at the end, I'll introduce a couple of trials that are ongoing right now in Angelman syndrome that are ASO trials. And so just basic biology, as you know, DNA gets translated into messenger RNA, which then gets transcribed into proteins that do all the work in our bodies and brains. ASOs are basically these kind of chemically modified strands of nucleotides that are that bind to the RNA. So they're complementary sequences to that messenger RNA. And when they bind to the RNA, they can basically stop or change translation of those genes into RNA and then into protein. And so they can directly affect, essentially, again, the translation of DNA. And so in that way can really affect the downstream effects of whatever the genetic change might be in an individual. Now, ASOs, they don't cross the blood-brain barrier the way they're delivered, so they do need to usually be given intrathecally. But ASOs have been really exciting in our field. And the example I always like to give, which I would say really blew the door open in child neurology, was an ASO treatment for spinal muscular atrophy, which is a really devastating neuromuscular condition that leads to death in infants who have type 1 SMA. They have a severe motor neuron atrophy where they essentially don't have motor function. And eventually they're unable to breathe and pass away. And with an ASO technology, there was, again, a trial conducted that actually was stopped halfway through the trial because they were showing profound gains, preservation of motor skills in these babies with SMA. And now it's led to this therapy called nusinersen that's been used now in thousands of children and adults worldwide. And actually, this was one of those examples in child neurology where I'll say, people often say like, oh, being a child neurologist must be so tough because you don't often have treatments. You're really good at diagnosing patients with conditions, but you don't really have treatments. And in many cases, that is sort of true, but a treatment like this for SMA really, really changed that. Sort of mentality, I think, in our field in many ways. It led to so much hope around the fact that even in these fairly devastating conditions, that we could actually find truly, disease-modifying therapies that changed outcomes profoundly. And so what's exciting is that same kind of ASO technology is now being tested in neurodevelopmental disorders. I showed this slide, and I'm going to tell you some details about these trials at the end, just of trials that are occurring at UCLA that are really precision therapeutics, right? So this is a trial it's a medication trial for epilepsy, but these are ASO trials in Angelman syndrome that are going to be launched later this year. So we are opening the door for these kinds of therapies to be available for these neurodevelopmental conditions. But here's the rub, and here's where I'm going to spend, we're at 12:05, so I'll spend about 15 minutes talking about this, and then again, introduce these trials and then open it up for questions. So we have the promise of these therapeutics, and again, all that, is credit to all these incredible teams of scientists who are really trying to test mechanisms of disease and then try to modify those with these kinds of therapies. But on our end, on the clinical end, we need to be ready in the sense that we need to be able to design and develop clinical trials that allow us to really determine if these treatments are actually going to work and how they're going to work. And so I think of that as clinical trial readiness. And I think this is a really important concept right now in our field. So just to get us all on the same page, remind all of us how trials work, right? So generally with a clinical trial, you have a group of patients, they get split up into two groups and in a random assortment. And so they're randomized to either getting the treatment or to getting either a placebo or maybe a wait list where they get the treatment in a staggered form. But there's some comparison group or time point that is there because you need to know if the intervention is better than not getting the intervention. And then you measure some sort of outcome, right? And for a trial to be considered successful, meaning for treatment to be considered successful, that outcome needs to be better in the group that received the treatment compared to the comparison group. Sounds pretty basic, right? Well, there's a lot of challenges actually in that basic design in these syndromic neurodevelopmental disorders. The first is that in these conditions where, as I said, this is not just straightforward autism. These kids have, and adults have, complex neurodevelopmental disabilities, often with rather significant intellectual disability and other challenges. Our standardized clinical measures often don't adequately capture function or change, right? So that outcome measure in itself is problematic. Second of all, we often don't have great measures to know whether the drug or the therapy is truly hitting whatever that target is, right? How do you know treatment has actually done what it's supposed to do even before you see a clinical outcome of interest? And then lastly, and this may seem very mundane, but it's a big issue, is that these conditions are rare, right? So, as I said, taken as a whole, 25% of patients, with a neurodevelopmental disability may have a genetic cause, but each individual syndrome is only, 1 in 10,000 patients. So these are small numbers. And so how do you do a trial with adequate sample sizes? And how do you get patients access to these kinds of trials when they're all over the country and actually all over the world? And so much of the work that we've been doing here at UCLA is to tackle those three challenges. And that's in these three different ways, right? So from the standpoint of the challenge around clinical endpoints, we've been doing work to try to figure out what are the most meaningful and measurable endpoints in these patients. Secondly, we've done a lot of work focusing on biomarkers, looking at mechanistic biomarkers that might be able to inform drug target engagement to tell us that a drug might be hitting a target. And then lastly, we've been working quite hard, and this is super relevant, especially now with COVID and all of the restrictions around COVID, is designing protocols and developing methods that maximize access and scalability, through remote assessment and remote delivery. So what I'm going to do, again, in really just 10 minutes, is show you examples of each of these. I'm not trying to be exhaustive by any means, and each of these can be a talk unto itself. But I want to show you each of these points just to, again, make a point, right? That we are doing work in this space, and that this is where the field is really positioning itself so that we can be successful as these trials are coming into fruition. So let's start with the first. And the syndrome that I'm going to talk about here is called Dup15q syndrome. Many of you have heard me talk about this condition before. These are kids and adults who have duplications in chromosome 15 in this region that's often called the Prader-Willi-Angelman critical region, because in kids who have Angelman or Prader-Willi, one of these genes, UB3A, is deleted or is not expressed adequately. Dup15q, though, encompasses a bunch of other genes as well, okay, in that region. What's important, though, clinically for you to know is that these kids and adults have what I like to consider kind of syndromic neurodevelopmental disabilities, meaning they have a genetic cause for their neurodevelopmental disability, but they have a lot of the challenges that we see in other genetic conditions. They have low tone and motor delays. They have global developmental delay. Most of them meet criteria for autism, and many of them have epilepsy. So in many ways, it's a good representative condition to be able to think about these problems around clinical endpoints. So we did a study, and it was a large-scale study funded by our IDDRC over several years, where we wanted to collect data on as many kids and adults with Dup15q syndrome to really ask the question, how do we assess developmental domains in these kids? What would be good clinical endpoints to be able to measure as we get ready for trials? And so what we did was we tested kids at UCLA, but we also went to family meetings that the patient advocacy group had organized. And this is, again, one of the power of these PAGs, is that they have family meetings. And so all the kids come there. What better place to go and engage families with research? So we did this at three different settings, and we tested both kids and adults, but we're just going to show you, I'm going to show you the data on the kids, which was recently published. And we looked at cognition, motor skills, adaptive skills, as well as social skills. So the first study that I'll talk about quickly, again, I show Charlotte DeStefano's picture here. She really spearheaded a lot of this work and is first author on the two main papers that came out of this study. In the first smaller study, what we did was we first compared a subset of these kids to IQ matched kids with what we call non-syndromic autism. So these are kids who have autism, often have other developmental challenges, but don't have Dup15q syndrome. Because we wanted to know, are there profiles that might be more specific to Dup15q? And what we found here is that, and not surprisingly based on our clinical impressions, is that our kids with Dup15q were significantly more challenged or delayed in motor skills. And this is both showing on the Vineland, which is a parent report, their gross and fine motor were delayed. This is looking at the Mullen, which is a direct assessment. We also here found that their gross motor skills were delayed, much more challenged or delayed compared to our non-syndromic autism group. So again, here's gross motor and here's fine motor. So again, motor skills significantly delayed. And this is actually interesting because this has been borne out in much larger cohort studies of syndromic autism. So this is a study that was done out of the Simon Simplex collection where they compared syndromic and non-syndromic autism. And what they found was one of the big differentiating factors here was age of walking. So age of walking was much higher or later in individuals who had a genetic condition. So, and again, there's many more studies. I like this one because it just shows it really clearly. So what we're finding in these motor delays is something that's quite consistent, maybe across other genetic syndromes as well. And that work actually has really inspired some beautiful ongoing work by Rajitha Wilson, who's in our autism center, who actually just recently got her K23 to look at motor skills in infants at risk for autism. But here, she actually used a more granular, really nice quantitative measure of motor function using a gait analysis software where kids walk across a mat. And by walking across the mat, you can actually generate really nice quantitative data on the child's gait. And you can see this is a gait map of Duke 15Q compared to a child with non-syndromic autism. And you can see differences. And what she found were there were differences in all kinds of aspects of the gait, including velocity, step length, other domains as well. And this was something that could be really nicely quantified. So this is a measure that we absolutely can use in future trials rather than relying on these standardized measures maybe of motor function. Then in our larger cohort, and this was, again, a lot of the data when we went around to all the different patient advocacy group meetings, we wanted to get data on a bigger cohort to just ask, how do our standardized measures behave in these kids? And we found a bunch of things. I'm not going to spend a lot of time on the slide in the interest of time, but I want to highlight a few key things. So this is looking at a combination of cognitive measures, mostly the Mullen scales of early learning. And what we found was that, not surprisingly, these kids in general, and this is just Duke 15Q overall, blue is kids without epilepsy and red is kids with epilepsy, we find that they've, on their standardized measures, their scores are pretty low, right? So they're really skewed towards the left, if you will. And I want to mention that here we actually developed, Charlotte worked really hard on this. We didn't actually just rely on the standardized scores. We created ratio IQ scores, which is really the child's age equivalence over their chronological age, to try to get a little bit more of a scatter and some granularity in their cognitive skills. But you still see that they're clustering quite low, right? So that really, again, makes us think about, well, is this really the best measure to use if we're trying to look at change over a short period of time? Maybe not. What's really interesting, though, is then we looked at adaptive skills using the Vineland, which is a parent interview. This is a measure that that industry loves. The pharmaceutical companies are using the Vineland quite a bit in their clinical trials. And really nicely, we found much better stratification in the Vineland in our patients across these different domains. And you can see that here in this histogram. So again, we see a really nice distribution suggesting that the Vineland might be a better measure than our direct assessment, cognitive assessments. The other big theme here that I want to highlight is that we see that our kids with epilepsy are more delayed or challenged than our kids without epilepsy. And I think that's important when we think about who we're including in our trials and how we might want to modify measures based on whether they have seizures or not. We also found that some of our standardized questionnaires that we ask parents, especially around comorbidities or social skills, like the social responsiveness scale, which some of you may be familiar with, or the child behavior checklist, they're problematic when we're assessing kids who may not have very much language or may have more severe intellectual disability. We're asking questions like, does your child give unusual or illogical reasons for doing things? Well, if a child doesn't talk, that's a very difficult question for a parent to answer. And so we found that these measures were not as helpful in this population, which again is important when we think about whether we want to use them in trials. So to summarize this piece, I'll just say that when we're thinking about the most meaningful and measurable clinical endpoints, I think we really need to be measuring motor skills. And again, I'm showing you Duke 15Q as our example population, partly because we're excited about having trials in this condition, but also that this could absolutely be relevant for other genetic conditions as well. So we need to measure motor skills both through standardized and more innovative quantitative methods like what Dr. Wilson and her lab are doing. We need more flexibility in cognitive assessments to prevent clustering at the floor, either with ratio IQs or maybe with other kinds of assessments. Our adaptive skills interview might provide more stratification. And then lastly, very importantly, is that our kids with epilepsy do have more developmental challenges, and we have to take that into account when we think about designing our trials and how we measure endpoints. Okay, so the second piece in clinical trial readiness is this issue about biomarkers, okay? And biomarkers is a whole topic unto itself. I have a whole talk on biomarkers. I'm going to spend 3 minutes on this, but I just want to remind all of you what a biomarker is. It's any characteristic that's objectively measured and evaluated as an indication of either a normal biological process or a pathogenic process, or as a response to treatment. there's lots of different methods we can use to assess biomarkers, right? In diabetes, we look at, blood glucose. In hypertension, we look at blood pressure. In medicine, we have lots of biomarkers. You can imagine that it's a much taller order to think about a biomarker in a neurodevelopmental condition. And so many people use different ways to measure brain function, because we know that fundamentally what's affected by these genetic changes is the way the brain is fundamentally wired, right? How the brain is functioning in early development. In my lab, we use EEG, electroencephalography, which is a really beautiful way to measure, in a very developmentally sensitive way, neural function in real time. And we know quite a bit at typical development about what EEG oscillatory patterns should look like over time, so that we can then identify changes that might herald a disruption or a change. So going back to Dup15q syndrome, what's pretty interesting is, and I'll be honest, it's part of the reason we got interested in this condition to begin with, was that there were some clinical reports that actually noted that these kids had, on clinical EEGs, like the ones done to monitor for epilepsy, that these kiddos had some differences on their EEG. For those of you who are not physiologists, don't worry if you don't appreciate this, but really these kids have these very high frequency oscillations that look like a pattern that we see when patients are taking GABAergic medications. So Ativan, Valium, benzodiazepines. When patients are taking positive allosteric modulators of GABA, they have this EEG pattern. So that's pretty interesting for us, because in Dup15q, there's several GABA receptor genes that are actually overexpressed. And so we really thought, wow, this might really be a biomarker of this condition. This might herald altered or abnormal GABA neurotransmission. And so again, imagine if we have drugs that are targeting this system, this might be a very interesting biomarker of target engagement. And so we set out to quantify this biomarker. And I won't go into all these details, but we take great pains and lengths to try to get good EEG data on our kiddos, including training nets and getting them desensitized, all sorts of things. We actually collected the EEG data both at UCLA, and we took a team to the family meeting and collected data there as well. And long story short, we were able to quantify this biomarker. This was beautiful work done by a former graduate student, Joel Froelich, and also an amazing statistician that we work with quite a bit, Damla Senturk. And what we found was that kids with Dup15q, you don't even have to know anything about EEG to appreciate that this heat map is different in Dup15q compared to typically developing kids and non-syndromic IQ-matched kids with autism. What we found was that these high frequency oscillations that are occurring in the beta band are significantly more prominent in Dup15q syndrome. And this is a power spectral density plot for those of you who like looking at those, where you can see that bump in beta in these kids. This is just a scatter plot showing the distribution of the beta oscillations in Dup15q compared to our comparison groups. Since then, and I don't have time to go into this today, we've done a lot of work testing the properties of this biomarker. We've established that it's pretty stable over time. It's scalable, meaning we can quantify it with clinical EEGs. It seems to differentiate kids with and without seizures, so it relates to phenotype in that way. There are some interesting data we have on sleep that I won't get into today. And then Joel did a really nice follow-up study where he looked at, through clinical data, mechanisms underlying this biomarker. And what I'll say, and that paper was just published earlier this year or last year, is that it seems as though that GABA receptor overexpression is necessary to have this high beta oscillations. Okay, so let me pull this piece together before going into the last piece, which will only be a few minutes, I promise, which is, let's take us back to this idea of precision health in Dup15q, right? So if we think about having a patient that we see in clinic who has some of the hallmark features of a syndromic neurodevelopmental disorder, and we identify that that patient through microarray testing has Dup15q, and we know that that affects GABA signaling in some ways, and we assess if this patient has this biomarker of these high beta oscillations on EEG, right? And then we've done a lot of work, again, thinking about their clinical symptomatology as we develop therapeutics that are targeting the mechanism. For instance, having drugs that are negative allosteric modulators of GABA, for instance, or even really direct gene modifying therapies. we can use that biomarker as a really robust possible measure of drug target engagement. If this drug is working, maybe those beta oscillations will be reduced even before we see a clinical outcome. And then when we have the trial, we'll know what our clinical outcomes should be. We're probably going to want to focus on motor skills and on adaptive skills because those behave really nicely in this population, maybe better than our standardized measures of IQ or social skills. We might even want to think about other physiological patterns as an outcome. So again, a lot of the work we've done has really informed the way we would think about how we would design a trial in this condition. Okay, so our little detour when I got bumped off, I think slowed us down a tiny bit, but I want to just quickly mention this last piece and then just tell you about a couple of trials that are going on at UCLA. And then I'll open it up for questions. If you're okay staying a couple minutes after, I'm happy to stay on to answer more questions. But the last big challenge in these rare disorders is the fact that these kiddos are all over the country and there's not that many of them. And we want to make sure that we give access to all these patients and families, right, to be involved with trials or even our clinical readiness studies before we have trials. And a great example of this has been our work in tuberous sclerosis. So I'm not going to get into the details of the biology of TSC by any means. What I will say is that it's an autosomal dominant condition that leads to a loss of function of these two genes, TSC1 and 2. It leads to pretty significant neurodevelopmental challenges as well as epilepsy, with autism diagnosed in more than 50% of these patients. What's important is that these babies are diagnosed often with TSC in utero because of either fetal MRI showing either subependymal giant cell tumors or cortical tubers in the brain, so either brain lesions or because they have cardiac lesions. That's the more common way that they're diagnosed in utero, through these benign tumors in their heart. So they're diagnosed at birth, and we know they're at very high risk for developing ASD or other neurodevelopmental challenges. And so we have for many years been studying these infants prospectively to ask, are there early predictors or markers of atypical development in TSC? And bottom line, we published a lot of this. What we found is that, in fact, yes, by 12 months of age, infants who develop autism are already showing red flags. They have delays in nonverbal cognition, they have atypical social communication skills, and there's actually EEG biomarkers that also seem to relate to later atypical development. So these data were important for us because, our goal was not just to predict, it's really to intervene. And so this really necessitated an early intervention trial, right, to help improve developmental outcomes. And so we developed this clinical trial called Jasper Early Intervention for TS. Jasper is a phenomenal behavioral intervention developed by my colleague, Connie Castree here at UCLA, that improves social communication skills in kids with autism. This was a trial that was designed in collaboration with folks with my collaborator, Chuck Nelson at Boston Children's and Elizabeth Teal at Mass General. And we were really excited about this study. This received quite a bit of attention in the media and the patient advocacy group because it was the first behavioral intervention trial in TSC. So we were excited and we thought we'd enroll tons of people. And in the first year, this is the map of enrollment, with yellow being those patients that were enrolled and purple being those that were interested but didn't enroll. So we were struggling after a year. We had 3 families in our trial, and I thought, oh my goodness, this is my first R01 and I'm gonna have to give it back, and we're not gonna be able to help any of these families because we can't get anyone in our trial. And you can imagine why, right? So get into the design, but it was a weekly behavioral intervention where they needed to come to UCLA or to Boston Children's to receive intervention. And this is what people were telling us. They said, it's really hard for us to come. We can't travel. We'd love to be able to do it, but right now it's basically not convenient for us. And one of the parents said to us, if virtual intervention ever becomes an option, we'd be interested. So we went back to the drawing board, and Connie Castree's team was incredible in revising the intervention. And I give great credit also to Carly Hyde, who is our research coordinator on the study, who really has spearheaded so much of this work. And the bottom line is we went back to the drawing board and completely revised the intervention delivery to be remote. Where we essentially provided parents with iPads where they could learn the intervention in person once, and then they go home and they practice every day. They record sessions on the iPad. The session then gets uploaded to UCLA Box, and the interventionists at the sites evaluate the video, and then they do a Zoom session with the parent to give them feedback. And then the parent goes back and practices more, and then next week they do it again. So basically, it's a parent-mediated intervention that now is all being done in the home. And in the interest of time, I can't show you the videos of how this, to share that with you guys if you want later. But the long story short of it is that this was our map, and within 8 months, this was our map of enrollment. So our enrollment exploded when we offered it up remotely. And this has been actually a really important message that we would like to tell others too, which is that we have to be flexible and think about ways to make these trials accessible for these families. We just wrote this up actually, and it was just published last month, this whole process and experience of delivering this intervention remotely. And so, to finish up that point, and then I promise I'm almost done, I think that, the really important point here in terms of designing protocols that maximize access is we need to partner with our families, listen to them about what the challenges are, and develop and innovate, right? And continue to innovate effective and feasible remote assessment and delivery strategies. I love this phrase, the perfect is the enemy of the good. And I think that what I mean by that is that, sometimes we get so worried about needing to do it perfectly, which usually does mean coming in person and following, rigorous protocols. But if that's going to undermine our ability to give access to families, then we have to go back to the drawing board and figure out what is good enough. What are the key features that we have to preserve to be able to ensure that families can enroll in our studies. Okay, so I'm basically done. I just want to introduce you to the next step. So I really talked to you about how excited we are about clinical trial readiness. We are in the epicenter of this right now. It's really exciting. So we have a bunch of studies being done right now. So I'm just going to literally blast these at you at the end, and you guys can email me, or when there's time, ask me questions. Charlotte DeStefano, who I showed you, did a lot of the work on the Duke 15Q study, now has a grant to do remote assessments in Duke 15Q using a toolkit, if you will, called PandaBox. And that's going to be really interesting, again, for us as we think about trials in Duke 15Q. We are a part of a clinical endpoint study that's funded by Roche in Angelman syndrome. And this is a study that's actually asking about clinical endpoints to be used for a trial. And what's important about this is that most of the assessments are done in the home. So there's sleep monitoring, there's EEG, there's parent diaries, and it's all done most of it's done remotely, again, with the goal of improving access and also thinking about the right endpoints to be used in trials. And then we have 3 trials. So right now we're in the middle of an epilepsy trial for 2 specific genetic conditions, Duke 15Q and CDKL5. This is a sort of an indirect GABA modulator that's being used for severe epileptic encephalopathies, and that's for kids ages 2 to 18. We just got approved to be a site. We'll probably roll out this fall. Roche actually has an ASO trial for Angelman syndrome, which is, again, is a neurodevelopmental syndrome, so quite similar to Duke 15Q in many ways. This is a phase 1 clinical trial of an ASO. It's monthly intrathecal injections of an ASO. And again, a lot of the work that we've done thinking about endpoints is being used as we think about the exploratory endpoints, at least in this study. And then another pharmaceutical company called Genetics has another ASO trial, also in Angelman syndrome, that we are also going to be a part of. That will probably roll out a little bit earlier than Roche, so probably end of the summer. And so again, we're in it right now. It's such an exciting time. And so I'm just going to end with this, right? So going back to this question, how will this testing help my child and family? I gave you the list of what I say to families, but I think above and beyond all of that, I really, really believe that our genetic testing is providing all of us, caregivers, clinicians, parents, families, all of us with hope and optimism for the future, that we can actually improve outcomes and really, really help these families and these kids live the best possible lives that they can live. And that to me is incredibly exciting. It's what gets me up in the morning to do this work. And so with that, I'm gonna stop and just thank my amazing group here. I'm sorry I went over by like 2 minutes, but I'm happy to stay on and answer questions. Liz, I don't know if you're gonna moderate the questions, but again, happy to answer them as they come. So thanks for listening.
Thank you, Dr. Jeste. That was an excellent and really interesting lecture. So much information. Thank you. We will actually stay on the call if you're okay with that, and moderate a few questions. Will Francis will be moderating the Q&A. For those of you that have to leave us, we understand, but we thank you for being on the call. But with that, we'll go ahead and open it up to questions.
Hi, great, thank you. So the first question is, what are some equity issues your team and many of the PAGs are seeing regarding access to genetics testing? And then the person makes a comment to help clarify this. I have worked with families who have been able to get this testing via Medi-Cal, but I wonder how referrals are occurring and what are the educational barriers many pediatricians or low-income clinics-based primary care doctors are facing. How can we move this forward, testing for all children, and what are the positive and negatives of such widespread testing?
Great question, and I wish I had a quick answer for you. What I will say is that there absolutely is disparity in testing, and I think part of that is, it's twofold. One is it's referrals and it's provider knowledge about the availability of testing. That first level of testing, though, the microarray, is actually now covered by almost all insurances. I mean, we still struggle sometimes with certain types of Medi-Cal, but really insurance is covering the microarray almost across the board at this point. The challenge and the barrier is actually in having the team to be able to help advocate for the insurance authorization and the testing. So our clinic is happy to help with that. I should emphasize that our caring clinic, we take essentially all insurances. We see a very wide range of families with all socioeconomic statuses, if you will, and backgrounds. So we are seeing a wide range, and we're doing our best to help facilitate that. You know, our geneticists at UCLA, I don't know if any of them are on this in this talk right now, but they're also great and are seeing, patients across different backgrounds and can help with the genetic counselors to advocate for testing. But I agree, it is not as widespread as it should be. And again, I think that because now we have actionable, we have actionable items that we can pursue with the testing, I firmly believe that we should be advocating to get that testing in everyone possible.
Next question is, thank you for that. Have you been able to look at the impact of family dynamics, particularly in families with parents who have syndromes such as Angelman, and how this impacts various domains of development?
I'm going to assume the question is about once a diagnosis is made, how does it impact? And it's a very interesting question. And we actually have been doing these diagnostic journey interviews with our families that is trying to capture some of that experience. What I will say anecdotally is I think there's a big range. I think for some families, having a clear, concrete cause and often a clear path of action forward can be comforting and can help them really have a good, solid sense of next steps. For other families, it can be very difficult because of the fact that in many of these conditions, the challenges are great, right? And the developmental delays and again, the sort of cognitive and behavioral challenges can be quite significant, and that can be sometimes overwhelming for families. So I've definitely seen the response vary. What I will say though is, especially with the power of these PAGs, what I often have found is that even if initially there is there's grief or just fear around the diagnosis, over time, as they develop that support network and find other families who have children who are facing some of the same both challenges and joys in their experience, that gives them such a great sense of community that it gives them, I think, a lot of comfort and again a really nice sort of clear path forward.
Great, thank you so much. There are only two more questions. One has to do with someone on the call wanted a clarification or an explanation of what Do15Q is. But before we do that one, let me insert this one. One problem with clinical trials focusing on autism and other disorders is the fact that heterogeneity in the sample can reproduce the apparent efficacy of the drug. What are your thoughts on how we should combat this?
Totally agree. And that's why, I showed you that precision health slide in the beginning, which was, we can tackle treatment trials in different ways. And one is that you just take the whole heterogeneous sample and just take a subset and try the drug or whatever the treatment is. And that's been really, that issue of heterogeneity is what has stymied success in large part in autism trials. So meaning when we take autism as a whole, which is, again, autism is defined not by a genetic etiology, but by a list of, behaviors and developmental features. If you take that whole spectrum and ask whether one drug or one treatment will help a whole group, the answer is probably not, right? So it'll probably help a subset, and it might be because there's variability in the cause and variability in the presentation. But there have been so many autism trials that have failed, partly because we've not been able to show that the group as a whole, the treatment group, is showing benefit compared to the placebo group or the control group, right? And so that heterogeneity is very problematic. That is why, though, that we think that these trials in these specific syndromes may be more likely to be successful, because we're dealing with a slightly less heterogeneous population, at least from the standpoint of biology. So they're more homogeneous in their underlying cause. So if you have a treatment that's targeting the cause, the hypothesis is that you would be able to see a change in more of those individuals. But that is something that we need to, keep trying to tackle. The heterogeneity issue is a big one.
Great. And then can you say some about Do15Q, what it is?
Yeah, so sorry, and I'm sorry I didn't spend more time on that, but so Do15Q is a copy number variant syndrome, which means that there is a piece of chromosome 15 that's duplicated. So there's either double the number, in some cases, sometimes kids have even triple the number of that piece of the chromosome on chromosome 15Q. There's about 30 genes in that region, many of which, because there's multiple copies, are overexpressed. And so that leads to the developmental features that I described. So these kids have global developmental delay, they have autism, they have epilepsy. It has been fairly well studied because of the fact that microarray has been around for quite some time. So we've been able to use that test to identify these kids. And because because it's so highly penetrant for developmental delay in autism, we've known about, Do15Q for a very long time. And so it's actually been considered one of the most common copy number variant syndromes associated with autism and neurodevelopmental disabilities. So again, it's just one of the many genetic syndromes that we study that, again, leads to a neurodevelopmental disability.
Well, thank you so much to Dr. Jeste for that clarification, for this very interesting and informative lecture on clinical trial readiness. I think we're unfortunately out of time. I do appreciate Dr. Jeste staying with us for an extra 10 minutes to answer some of those questions. Also want to thank Dr. Jeste for her flexibility in rescheduling this lecture for our secured Zoom call. Thank you to all of our attendees for joining the meeting today, and we wish you and your families safe and healthy. And we look forward to seeing you all at our next Tarjan lecture. So thank you so much. Have a good rest of your day. Thank you, Dr. Jeste.
Thank you, everyone. Bye-bye.